Okay, so the company PDC Line Pharma has been established in 2014 as a spin-off from the French Blood Bank in France, based on the work of the scientific founder Joël Plumat, that has discovered in the blood of leukemia patients a very unique population of cells. In fact, the leukemia was affecting the plasmacytoid part of the dendritic cells of the patients. And that has been done 15 years ago. And based on the blood of his patient, the PBMC has been collected, and it generated a GMP cell line and a master cell bank, GMP approved by the French authorities, which is still the substrate we are using today as a vaccine to treat patients. So today we're a team of 20 people, including seasonal executives from biotech and pharma. In terms of funding, we get 17 million euros so far in equity and non-dilutive money from the Walloon regions. And we are in the process of raising a new round of financing of 11 million euros. So the technology, the PDC line, PDC is for plasmacytoid dendritic cells, is a cell line. So it's today the unique off-the-shelf and highly scalable cell line, which is of plasmacytoid origin, and the plasmacytoid phenotype of the cell provide very interesting properties. I will go back to that. It has been infused already 9 times in 9 patients several times, and we have observed no rejections of the cell line. So it's an allogenic cell line, of course, by definitions, but has been infused several times to the patients, and we have never observed any rejections against the HLA of the cell line or through MLR, any activations of the allogenic CD8. It's a clinical stage company because we are initiating in this month, in fact, a phase 1b/2 for non-small cell lung cancer. We did already an academic clinical trials in melanoma in 9 patients. That is closed. We are focusing now in non-small cell lung cancer, where the unmet need is bigger, and we have in development program in neoantigens in collaborations with some partners. We signed very recently a licensing deal with a big Korean company, LG Chem, licensing in Asia. LG Chem is one of the biggest chemical company in the world. I think it's the 10th and the lead and the number 1 in Korea. And they have a life science group that is going to develop in clinical development, and we hope in the future commercialize the product in Asia. The total deal value for PDC lung, the lung part, is 108 million euros plus significant tied royalties on net sale in Asia. And we hope that new opportunity will come again in the next future. Let's go now in the positioning of the product. So the checkpoint inhibitor has really been a game changer in immunotherapy, in the cancer immunotherapy. We have now a quite clear understanding of the mechanism of defense of the tumor on the CD8. We know that the CD8, the cytotoxic CD8 that have the role to kill cancer tumor cells that recognize specific antigens, that are presented by the dendritic cells to the T cells. These mechanisms, which is natural mechanisms, is completely inhibited by checkpoints that has produced by the tumor, by the whole tumor, that completely lock the system and avoid the activations of the CD8 and the killing process. So this checkpoint inhibitor inhibits these mechanisms has provided a big value, of course, for the patients. But we observe that not all patients respond to checkpoint inhibitor due to inefficient pre-existing immunity. In fact, even if you remove the brake, you still have the lack of immunity of CD8 activations to have an effective anti-cytotoxic activities. You see here the response rate, melanoma 33%, lung 44%, and colon MSI 40%. And we believe that association of checkpoint inhibitor with cancer vaccine is one of the future positioning to help patients, associations to help patients, and to get even better response rates. So complementary activation of anti-tumor T cells by cancer vaccine can boost anti-PD-L1 efficacy. So first generation of vaccine has been developed in the past. We know the dendritic cell-based vaccine, the autologous cell-based that has been developed, the antigen-based vaccine. But all this approach so far were not very successful, even if success for some of them, some product has been on the market, but they have not been successful because the lack of immune suppression, due to the presence of immune suppression, all the first generation of products were done before, were developed before the arrival of checkpoint inhibitor. And secondly, because all of them presented some specific issues, like the allogenic, the autologous dendritic cells, that have manufacturing and cost of goods issue, and heterogeneity, the batch-to-batch consistency with autologous dendritic-based vaccine is a big issue. And the antigen-based vaccine, the approach that GSK and Merck and others have developed, had some issue related to the targeting and the immune adjuvant related. We believe we can tackle these issues. First of it, why? Because first of it, we have an allogenic cell line. What it means that with a master cell bank approved by the French authorities, we just have to take one sample of this master cell bank, and in bioreactor, because we have not developed a single-use bioreactor, we can produce billion and billion of cells in just 9 to 10 days. And these cells can grow in a synthetic medium without any calf serum, without any interleukin, GMP interleukin or cytokine to activate, to maturate the cell line. And at the end of the manufacturing process, we just have to load the cell line with peptide, GMP peptide representative of the tumor that we want to target. And this GMP peptide stick to the HLA present in the surface of the cell line. Then we irradiate the cells, because once again, it's a leukemia cell line coming from the patient, the leukemia patients, to stop the proliferations. Then we are using the aseptic technology to preserve the cells that can be stored in liquid nitrogens. And when the patient is ready, we thaw the cells and infuse to the patients. So this process is ongoing. It has been approved. We are GMP approved for the QC and batch release, and the manufacturing is still done today by the French institution, the French Blood Bank. We have been ATMP classified 2 years ago. Second key advantage we provide with the cell line outside the consistency and the cost of good, because once again, with the process I've presented to you, we are 1 to 2 log lower in terms of cost of good with the autologous approach. So we did different benchmark study to compare the PDC line loaded with one specific antigen, the melanin, with autologous and allogenic conventional DC, the myeloid DC, that has been published, what I'm presenting here. And we follow by fax dextramer with the presence of PBMC of donor, the expansions of the cytotoxic CD8 T cells specific of the melanin. And as you can see here, the fold increase is a lot bigger in our case. We are 20 to 200-fold more potent than the classical DC. Why? That is related to a unique mechanism of actions that has been, we believe, in big part elucidated by the scientific founder over the last 15 years. We know that the cell line has a receptor to HLA class 2. They can activate the CD4. And the CD4 are the ones that activate the allogenic rejection. So, but we need as well the IL-2 to activate the CD8. We know that the cell line produces, of course, HLA-A2 and the complex peptide HLA. And there is a co-stimulatory molecule present in the cell line. And these three pathways are the natural free pathway required for the CD8. So for the moment, everything is normal. So what are the new things? First of it, the lack of allogenicity is related to the lack of some co-stimulatory molecule, like the CD480 and CD86, and this lack induces what we call an abortive expansion. So the CD4 are activated, but they do not multiply, and they are not in quantity enough to induce rejections against the cell line as it. So that's for abortive expansions and the lack of allogenicity. Why this big boost of CD8? That is related to a fourth mechanism of actions involving the monocyte, the circulating monocyte, that are activated as well by the CD4, that produce a specific interleukin. We put IL-6, X, which is transpresented to the CD8, and this mechanism is the fourth mechanism that induces the huge expansion of specific memory CD8 T cells. And last but not least, we know that the cell line is negative to PDL1 and PDL2, so there is no break. So when the cell is alive, it just activates. There is no inhibition potential. And this fourth pathway activates very strongly the cytotoxic CD8 present in the blood of the patients. So everything I'm showing you is not published yet, has been done ex vivo in the blood of the patients. What is important as well to underline is that the cell line is coming from a Caucasian patient, so it's HLA-A2 restricted. It's the majority HLA present on the membrane. But we know that we can modify genetically the cell line to produce additional HLA that are more prevalent in Asia, like A24. We did feasibility study, and we know it works. We can present specific HLA A24 peptide to the CD8 and with activation of cytotoxic CD8. So we can modify genetically the cell line to produce additional HLA, and we can as well, instead of loading with GMP peptide, modify genetically the cells to produce endogenously polypeptide. We can as well use RNA, electroporated through electroporation technique, RNA, to present the peptide through the HLA of the cell. So the platform is very versatile, can be modified very, very easily, and we can develop in the future GMP master cell bank with modified cell line. Is something we have in the plan. We did several studies in animals. I'm just showing one of them here, where to show the effect on a tumor, graft tumor in humanized cells. So we have humanized the mouse with PBMC of a healthy donor. Then we have infused HLA-A2 and melanoma-positive tumor, aggressive tumor. And after that, we have vaccinated 3 times the mouse with a positive and negative control. Positive is PDC line with melanate, negative is PDC line plus flu. And as you can see, the growth of the tumor in the mouse is extremely high with the negative control. With the positive control with PDC line and melanate, which is expressed in the tumor and infused to the cell, to the mouse, you see that we completely blocked the proliferation of the tumor. What has been done as well is at the end, before sacrificing the mouse, or just after sacrificing the mouse, we took biopsy of the tumor in the mouse and look for the TIL, the tumor-infiltrating T cells, and the ones that are melanate positive. And we observe that 11% of all the TIL in the tumor were melanate positive, which is a huge number, meaning that in these models, the cell line has been able to prime and expand CD8 T cells that have migrated to the tumor microenvironment to make the job, which is to kill the tumor. That has been published as well in 2010. We did study as well, feasibility study in neoantigens. It's a very hot topic currently, and we wanted to show that the cell line can as well present, prime, and expand CD8 T cells specific of neoantigens. For that, we have taken 2 mutated peptides that has been described in the literature. You see here the references where the mutation is clearly established. And we have taken the PBMC of the blood of 3 healthy donors to see if we can expand when we load the cell line with the mutated peptide, the CD8 specific that is done by FACS, the CD8 specific of each of these peptides. And as you can see, the answer is yes. We have 150-fold increase in just 20 days, which is extremely quick and easy in our case, and that with a purified CD8 of healthy donor. In this context, the quantity, the clone of CD8 specific of this peptide is extremely low. It's less than one out of 1 million. So it shows you the potency and the power of the cell line. So as I said in the beginning, we want synergy with checkpoint inhibitors. So we have demonstrated this synergetic effect ex vivo. We have taken the blood of melanoma patients, 12 melanoma patients, and in melanoma, which is a very hot tumor, we have a lot of checkpoint present in the blood, so a lot inhibitions, tumor inhibitions. So what we did, we take the cell line with, once again, the melanate, baseline and PDC vac with melanin, and we look after 1 week the expansions. So we have, when we take the cell line alone, 50-fold increase of expansion. After 1 week with just the checkpoint inhibitor, no cell line, we have almost nothing, 2, which is not statistically significant. But if we combine both, checkpoint and vaccine, we observe a very strong synergetic effect. That has been done in a cohort of 12 melanoma patients with at least 2-fold increase. So we are in the range of 35. So we have shown here, ex vivo, that we are able to provide synergetic effect with checkpoint inhibitor. Next clinical trials, we will do it with checkpoint inhibitor. The first one, the melanoma clinical trial that I'm showing you here, has been done without checkpoint inhibitor because it was an academic trial started before the approval of all these products. It has been done on 9 patients with advanced melanoma with the cell line pulse with 4 shared antigens that you see here. 3 times, we have infused 3 times to each patient. Results were promising. We have acceptable tolerability and safety profile comparable to the other therapeutic vaccine. We have demonstrated no allergenic rejections, no antibody against the HLA of the cell lines, and through MLR, we never seen any activations. And last point that was extremely important as well, we have observed significant antigen-specific CD8 T cell response in the blood of these patients, directly detectable without expansions, in vitro expansions. And that was extremely important because we have demonstrated that in human, the vaccine works. And that despite only 3 infusions, in the next clinical trial we infuse 6 times, and no associations of checkpoint inhibitors. Next clinical trial that we are starting now is on 66 patients in non-small cell lung cancer without and with checkpoint inhibitor, with objective to follow the safety again, immunological response, response rate, and the PFS. So that is starting now, and the next one will be a double-blind randomized controlled study to confirm the clinical activity we want to observe in the first clinical trials. So in terms of development plan and funding strategy, as I said, we would like to get 11 million euros of capitals at the top of the non-dilutive money we get so far to fully finance these clinical trials and to carry on the work on the platform, to work on neoantigens and multi-HLA, etc. And after that, we will expect to do another fundraising in the range of 30 million euros. And that's my last slide, and I would like to thank you for your attention.